हेलो स्टूडेंट्स, तो आज मैं आपके लिए अपने चैनल बायोसिम्प्लीफाइड पे क्लास 12 का एक चैप्टर पे वीडियो लेके आई हूँ। आप वीडियो को अंत तक ज़रूर देखिएगा और इस चैप्टर पे मैं बहुत सारे वीडियोस बनाने वाली हूँ, दैट इज़ ऑन जेनेटिक्स। सो लेट अस स्टार्ट विद प्रिंसिपल्स ऑफ इनहेरिटेंस एंड वेरिएशन। तो जो इस चैप्टर का फर्स्ट पार्ट है बच्चों, उसमें आपको एकदम बेसिक अंडरस्टैंडिंग आपको चाहिए जेनेटिक्स के बारे में, जो आपने क्लास 10 में पढ़ा हुआ है। ठीक है, तो क्लास 10 में आपने शायद पढ़ा होगा बहुत सारे टर्म्स दैट इज रिलेटेड टू जेनेटिक्स लाइक वेरिएशन, इनहेरिटेंस, होमोलॉगस क्रोमोसोम। तो एक बार हम उसको रिपीट करेंगे। तो व्हाट इज जेनेटिक्स? जेनेटिक्स इज द ब्रांच ऑफ साइंस दैट डील्स विद द इनहेरिटेंस एंड वेरिएशन। दो टर्म यहाँ पे है, आप देख सकते हो, इनहेरिटेंस एंड वेरिएशन। ओके, so what is this inheritance and variation? Now inheritance is the process by which the features or the characters transmitted from parent to offspring. Okay, and this entire study of this inheritance or transmission of feature is known as heredity. But when the features are transmitted from the parent to the children are not the exact copy of their parents. Some differences are there. Differences between the parent and the offspring and difference between the siblings, right? So this differences is known as variation. Variation can be in term of physiology, morphology, or in the behavior characteristics, right? So moving on to other terms related to genetics, सबसे पहले आ जाते हैं homologous chromosomes, right? So what is this homologous chromosome? Homologous chromosomes are the chromosomes in which both the chromosome acquire same shape and size. Okay, so they have same type of shape and their centromere position is also same. So this is called homologous chromosome. Now in human body you all have studied that there are 23 pairs of chromosome, right? Total 46 in each somatic cell. So out of 23, the 22 pairs of chromosome are involved in maintaining our body features. So those chromosomes are called autosomes. But the last pair or the 23rd pair contains gene that helps to determine the sex of the offspring, and that is known as sex chromosome. So in human body all the somatic cell contains first to 22nd pair chromosome called autosome, and the 23rd pair may be XX in female and XY in male is called sex chromosome. Okay, now what is gene and allele? Now gene, you all have studied, gene is the unit of heredity, right? So what is this actually this gene contains? This gene contains specific nucleotide sequence in the DNA. Okay, so those specific nucleotide sequences in DNA is known as gene. They code for particular protein, and for each gene there is a alternative form which is known as allele. Clear? So suppose capital T is the gene for tallness, so alternative form will be small t. Now moving on to next term, that is genotype. The genetic constitution or makeup of an organism is called genotype, but we cannot see the genotype of an offspring, right student? Right, whatever features you have, those features which can be observed, it is called phenotype, but genetic constitution you cannot observe. So phenotype is the features which are observed with respect to morphology and physiology, right? Next, moving on to what is this homozygous and heterozygous condition. Now homozygous condition means the same allele pair is present. Same allele pair will be present. For example, if I write capital T and capital T, it is homozygous condition. But if I write capital T and small t, this is heterozygous condition. So homozygous means in the pair same alleles are present, and heterozygous is the condition where two different alleles for a character is present, right? So these are all about the specific genetic term. I want to include two more terms here, dominant and recessive. I hope you remember. The dominant gene is the gene which is expressed, okay, and another one is recessive. Recessive gene is the gene which is suppressed. So if you see in this condition, capital T, this one heterozygous condition, capital T is the dominant gene and small t is the recessive. Capital T will be is a super ruling gene, but small t is suppressed by the dominant gene. Moving on to next topic, that is about the inheritance, law of inheritance. Now you all have studied that Gregor Johann Mendel, he had conducted several experiments, hybridization experiments on garden pea, right? I think you remember that. So garden pea, its scientific name is Pisum sativum, and for 7 years he continuously he was doing several experiments, and he proposed certain laws on inheritance pattern in the living organism. But question is, why Mendel had selected only garden pea for his experiment? Now some of the points I have written here, you can go through those points. Now this garden pea is an annual plant which survived for only 1 year, so several generations can be studied. Then flowers are complete bisexual, they undergo self pollination. Then self pollination can be prevented and cross pollination can be done, okay. And it is found, Mendel found, there are several different contrasting characters in the garden pea, and this garden pea in one generation has a capability to produce huge number of seeds. After performing several experiments, here this Mendel's work was unrecognized for several years, but when several scientists, they have taken the principle of this law of Mendel, they also finally concluded that whatever laws were proposed by Mendel was more or less correct. Now what are the reasons behind this Mendel's success? Now Mendel, whenever he was performing experiment, he used to study only one character at a time, okay, not several characters, one characters. Then he carried out experiment up to 2 to 3 generation, okay, second and third generation, and he whatever conclusions were given by Mendel on his experiment, those were not only based on biological logics but also based on mathematical and statistical analysis, okay. Then crossbreeding was done, pure varieties, pure lines of parent plants were ascertained by series of self-crossing tests between progeny of several generations, and then Mendel experiments dealt with large sample size. So these are the points which you can study under reasons of Mendel's success. Now next topic we'll discuss what are the different characters observed by Mendel in garden pea, in the garden pea plant. So this is the table showing the 7 different characters. You can see it is written dominant trait, recessive trait, okay. Now under dominant trait, round, yellow, green, full, these are the dominant trait. So if you see one character, shape of the seed, so under that dominant character is the round shape of the seed, and the recessive character is the wrinkled shape of the seed, okay. And if you see color of the seed, color yellow is dominant and green is recessive. Color pod, if you see, green is dominant and yellow is recessive. So in this way 7 different characters with their alternative traits were observed, and he had done two very important crossings, that is monohybrid cross and dihybrid cross. You have studied in class 10. Monohybrid cross is the cross by considering only one pair of contrasting character. So if I select one character, that is height of the plant, then two different traits are there, that is tall and dwarf. Okay student, so tall gene is represented by capital T and dwarf gene is represented by small t. Now two parents are taken, two parent plant with homozygous condition, you can see here, that is capital T, small t, small t. Gametes are formed, and after fertilization, this is the offspring form which has capital T, small t as the genotype. So when this F1 generation offspring are undergoing self crossing, you can see, then these are the possible combination of gametes. You know that in gametes the number of chromosome reduced to half. Then in F2 generation, this is the possible combination of offsprings. So homozygous tall 1, heterozygous tall 2 and homozygous dwarf 1. So if you see the phenotypic ratio, how many tall plants are obtained? Total 3 tall plants are obtained and only 1 dwarf plant. Okay. And genotypic ratio means how many homozygous tall? 1, heterozygous tall heterozygous tall 2 and homozygous dwarf is 1. So ratio is 1:2:1. Now moving on to second crossing, that is dihybrid cross. Now in dihybrid cross, two pairs of contrasting characters are taken. Now what are the two pairs of contrasting character here? One is color of seed. Okay, two different traits are there for color of the seed. One is yellow, denoted by capital Y, and another one is green, denoted by small y. Second one is taken as shape of seed. So for this, two different traits are there. One is round, denoted by capital R, and another one is wrinkled, denoted by small r. So you can see homozygous parents are taken, capital Y, capital Y, capital R, capital R. So this is the gamete, capital Y, capital R and small y, small r. After fertilization, the F1 generation offspring, they have heterozygous condition, capital Y, small y, capital R, small r. For second generation, self pollination is done between capital Y, small y, capital R, small r, and 4 different possible combination of gametes were obtained. Okay, what are the 4 different combination? Capital Y, capital R, capital Y, small r, small y, capital R, and small y, small r. So this were crossed, and finally after fertilization 16 different types of offsprings were obtained. You can see. So if you notice students, yellow round total 9 are there, green round total 3, yellow wrinkle total 3 and green wrinkle total 1. So phenotypic ratio is 9:3:3:1 and genotypic ratio is bit complicated, you can see here. So these two crossings are the basis of law of inheritance. So in next video I will discuss about the law of inheritance. I think concept is clear. You have recalled everything whatever you have studied in class 10. So you share my channel, like it, and please subscribe it. Share it with your friends also because they will also get benefited. और मैं इस चैनल पे इस चैप्टर पे और भी सारे वीडियोस डालूँगी, आप ज़रूर देखिएगा. थैंक यू सो मच.